introduction, if you like, focus coming down on time. Okay? Alright, so let's just jump straight in. Neurocontrol exercise and muscle. You should pretty much all know about organisation of the nervous system. Okay? Obviously have the central nervous system, which is made up of the peripheral nervous system, made up of the sensory and the motor division. Okay? In terms of the motor division, so the peripheral nerves, we have the autonomic nervous system and the somatic nervous system. What's the somatic nervous system? Do you remember?
Things that are like you move yourself.
Yeah, so it's sending information out to the muscles. Okay? To the muscles, skeletal muscles, for instance. Okay, somatic nervous system. The autonomic nervous system, okay, made up of parasympathetic and sympathetic. It's involuntary, as you know. Okay, we'll talk about briefly coming up. So that's our overview of the nervous system. In terms, again, you should have seen something like this before in previous classes, a neuron. Okay, it's a cell, obviously. Okay, we've got our nucleus, our cell body, we've got our axon hillock, which we'll talk about, becomes important. Okay, we've got the axon. Okay, and then we've got the axon terminals. We've got myelin surrounding the axon. The nodes around the air we'll talk about, they become important to us, and obviously again with the cell body we have these dendrites as well, which become important. Okay, who can remind me of what the importance of dendrites? Synapses with another neuron, so it receives information from another neuron. Okay, if you recall. Okay, receives information. I am going to talk about the myelin sheath again coming up. I'm going to talk about those around the A, and obviously the axon terminals, so that can go here, all the end branches that come into this. Okay, so I'm just going to save that for a second. The nerve impulse we actually call the action potential, right? The electrical charge that propagates down the axon. Okay, so we know for sure that the actual action potential is generated, you recall, by the axon hillock and propagates down the length of the axon, right? You remember, to the axon terminal. Okay? So, nerve impulse or action potential. Resting membrane potential. Okay? What is some of the resting membrane potential for me really briefly in a cell, in a neuron? So at rest in the neuron, in a cell, we have an electrical charge inside the cell of a neuron of around about minus 70 to minus 90 millivolts. Okay? It's negative. That's our resting membrane potential. Okay, that's our resting membrane potential. The difference between electrical charges inside and outside the cell caused by separation of charge across the cell membrane. That's our cell membrane right here. Okay. Okay. In terms of concentrations of ions inside and outside the cell, okay, there's a high concentration of sodium outside the cell compared to inside the cell, right? So that's my concentration there, sodium, and there's a small concentration inside the cell, and that reverses with potassium. Okay? Or potassium inside the cell and outside the cell. Okay? Now remember, ions flow down a concentration gradient, if you recall. Okay? So sodium's going to travel in that direction. Potassium's going to travel in that direction. Right? So you've got a positive charge coming in, you've got a positive charge leaving. This guy here that I've drawn, this circle, sodium, potassium, ATPase, or otherwise known also as the ATP, the sodium potassium pump. Okay? What's the purpose of the pump? What's the purpose of the pump? So it's obviously to do with sodium and potassium. Moves them in and out, you're quite right. It's actually also termed an ATPase.
Is it facilitate diffusion? Is it diffusion of it in and out, so it needs something to be sent in and out, like you can't do it actively?
Kind of. You're kind of going on the right track.
So it's moving the sodium and potassium against their concentration gradient.
Yeah, exactly. It's moving potassium and sodium against their concentration gradients. So it's using energy, it's using ATP to drive that. Okay? ATPase. It's an enzyme that's going to split ATP into ADP and inorganic phosphate. Okay? So in fact what you've got 3 sodiums coming in, 2 potassiums. What, the one going? Yeah, that's right. 2 potassiums coming in. Right? 3 sodiums moving out, 2 potassiums coming in. So the potassiums are coming in against their concentration gradient, right? Because they're usually going out that way. And the reverse of sodium. Okay? So it's taking more of a positive charge, just because it's 3 to 2 out. Okay? And also you've got a lot of, the structure of a cell is made up of a lot of protein, right? The cytoskeleton's all protein. And those proteins, or those anions, are all negatively charged. So they're holding on to a negative charge. So what with the sodium potassium ATPase, all these proteins holding on to a negative charge keeps the resting membrane potential very negative, around -79 to -90 millivolts. Okay? In essence, that's what's been said. Okay? Can it change? Of course it can change. Okay, absolutely. Okay, that charge can change. Okay, if we depolarize the cell, that occurs when the inside of the cell becomes less negative or more positive. Okay, so we're driving it more positive. We can depolarize the cell. We can hyperpolarize it where it becomes more negative. We can drive it more negative, say down to -150. You'll see this in a second. Depolarization, hyperpolarization, gradient potentials are localized changes in membrane potential. They're either depolarization or hyperpolarization. We'll talk about those coming up. And of course, action potential, which you all heard of before. Okay? So you're going from, say, -70 millivolts up to +30. You fire the action potential, and then you drop back down to the resting membrane potential of -70 millivolts. So yeah, we can absolutely change the resting membrane potential. Okay? Okay, again, you probably should have seen this, 30 to take 15, an action potential. Okay? All action potentials begin as gradient potential. Okay? They're summed, if you recall. Okay? For that, the depolarization to kick off, basically, you've got to reach threshold potential. Okay? So, in this instance, here is our cursor. Here's my resting membrane potential right here. It hasn't got a value, it could be -70 millivolts. There's the threshold. That threshold may be say stuck at -30 or -20 millivolts, right? For that action potential to fire, you have to reach threshold. So these gradient potentials are summed. If it reaches threshold, you'll heard of all or none, right? It will fire. You'll depolarize, become very positive. Okay, that's your depolarization state, and then you'll reach the peak, and then you'll repolarize back down to the resting membrane potential, but actually further than the resting membrane potential, hyperpolarize, and then come back to the resting membrane potential. Okay. Just a quick word on refractory periods. You've got an absolute refractory period and a relative refractory period. So basically, with the absolute, when a given segment of an axon is generating action potential, its sodium gates, we'll talk about those in a second, are open and is unable to respond to another stimulus. You can't fire another action potential. Okay? relative refractory period when the sodium gates are closed, the potassium gates are open, and repolarization is occurring. The segment of the axon can respond to the new stimulus. So you can fire another action potential. But immediately behind the gates that are opening, we'll talk about those in a second, the sodium gates in particular, you cannot fire another action potential. Okay, coming up. So, with the events during an action potential, then you have the resting state, this guy right here. You have repolarization, the propagation of the action potential down the neuron from the axon hillock. Then it goes back to resting, right? Repolarization, returning to the resting state to help on sodium potassium pump or ATPase. So what we mean is, there's your sodium gates, right? If your sodium gates are open, you bring the positive charge in. If you bring a positive charge in, you're going to move this towards threshold. It's connected, isn't it? Okay? That's the example there. You might have moved up to like plus 30 millivolts. You're going to fire an action potential. And the action potential then is propagating down the neuron in that direction. Okay? So you've got increased sodium permeability, meaning that simply means sodium coming in through this channel. Okay? Now it propagates and rolls down, okay? Rolls down the neuron. So we've got going on here. Okay. And what occurs behind, in terms of repolarization, behind the sodium channel is that the potassium channels then become open, and they're going to take the positive charge out. So then sodium gates close, you've got here, and the potassium gate is open. And that continually occurs down the neuron, right? It rolls down the neuron. Okay. So this looks something like this. There's our resting membrane potential, minus 70 millivolts, right? And minus 50 is our threshold. If you reach threshold, you'll fire the action potential. So 4, during depolarization here, this is when your sodium gates or your sodium channels are open. And you're bringing a positive charge into the cell, firing action potential. 6, okay, 6 is when your potassium channels are open and your sodium channels are closed. So 4, depolarization, sodium gates open, potassium channels closed. 6, repolarization, sodium gates closed, potassium gates open. Okay. Everyone okay with that? Okay. The velocity of an action potential depends on a certain factors. Myelinated fibers. You saw that myelin before that wraps around the neuron. Okay? There's gaps in the myelin sheath. Those gaps are called nodes of Ranvier. Okay? So, if you recall, there's our axon, okay? There's our myelin sheath wrapping around the axon. And at these nodes of Ranvier, this is where all our channels are located. Our sodium, potassium, there's actually, I've mentioned it before, there's chloride channels as well, there's calcium channels, all located at the nodes of Ranvier. And the action potential jumps from each node of Ranvier to the other. Because remember, there are some neurons that are unmyelinated in the nervous system. And their action potentials are very slow compared to axons that have this myelin sheath, and the action potential can jump from each node of Ranvier to the other. Okay? So they become important nodes of Ranvier, only found obviously in myelinated neurons. Okay? The diameter of the neuron also plays a part as well. The larger the diameter of the neuron, the faster the action potential, because there's less resistance to flow. So for instance, a fast twitch muscle fiber has a larger diameter neuron compared to a slow twitch muscle fiber, and that would make sense. Fast twitch, okay, rapid force, okay. So large diameter neurons, which in our case would be the fast twitch muscle fibers that come up early Okay, there's the key points. I'm not going to go through those. They're there for you. Or, I'm reading. Just to show you that we're not making up these nodes around the A. This is actually an electron microscopic shot here of some neurons. And you can see here, here's a neuron here. There's a node around the A. Here's another one there. There's another one right there. Looks like they're being pinched. That's where the channels are located. Okay? Nice shot. And obviously in a schematic, there it is. Okay? That's the real thing. Synapse. Synapse means obviously synapse between two neurons, or a neuron synapsing onto a muscle, neuromuscular junction. Synapse. Okay, we've heard that before. Impulse travels to a presynaptic axon terminal, where it causes synaptic vesicles on the terminal. We'll look at this in a second. On the terminal to release neurotransmitters in the synaptic cleft. And then we have a postsynaptic event, which we'll talk about. Okay. So a typical chemical synapse, right? So in this case, what we got, here's obviously our neuron, our axon. We've got the action potential of nerve impulse coming down. Okay. We've got neurotransmitters in vesicles. Synaptic vesicles contain neurotransmitters. This is our presynaptic side. And here's our postsynaptic side. So we've got our presynaptic side, we've got our synaptic cleft, synaptic cleft right here, or a space, and then we've got our postsynaptic ion channels right here. Okay, so what occurs? Well, we've got the action potential propagating down the neuron. Those vesicles will only adhere to this presynaptic membrane if we have entry of calcium coming in as well. Okay. So the entry of calcium, the action potential, and actually quite a few other factors which I don't have to worry about in this class, that causes these vesicles containing neurotransmitters to adhere to the presynaptic membrane and exocytose neurotransmitters into this space, into the synaptic cleft. Okay. The fate of the neurotransmitter then is to bind to these receptors on the postsynaptic side. So for instance, here's a blown up diagram. You've got the neurotransmitter, the green guy here. It's going to bind to a receptor that's physically bound to an ion channel. So this is what we call a chemically gated ion channel, or you can probably know the term a ligand gated ion channel, same thing. Okay? And what happens is the neurotransmitter binds to the receptor, and that opens up the sodium channel so sodium can come rushing in. Okay? Once it's done that, we don't need the sodium inside the cell anymore. The neurotransmitter can be degraded or recycled and used again. This is another nice shot here, actual shot of an electron microscopic shot of a neuron. It's a cross-section of a neuron. So again, you know that we're not making this stuff up, okay? So here we have the axon, we're looking down the axon. We have a lot of mitochondria. We're going to be talking about mitochondria coming up. You all know about mitochondria, powerhouses of cells, okay? Very, very important. There's a lot of them. As you can imagine, a neuron is a highly metabolic cell. It requires a lot of energy, okay? Here are our here are our vesicles containing our neurotransmitters. We have a Schwann cell around the neuron. The Schwann cell actually makes the myelin. Okay, so Schwann cell actually makes the myelin. And we can also see a lot of granules here. These are glycogen granules. Okay, these are glycogen granules, the storage form of glucose. Again, it's a highly metabolic cell that requires glycogen. Okay? Break down glycogen for it. Alright, you've probably heard the term before, the neuromuscular junction. Okay, this is where a neuron, an alpha motor neuron, coming from the peripheral nervous system, is going to synapse onto skeletal muscle. Okay? We've just seen the neurotransmitters that are released into the synaptic cleft. Okay? And the binding of the neurotransmitter causes depolarization. Okay, on the postsynaptic side. And as you know, once the threshold is reached, you try another action potential. Okay? And then that action potential obviously moves down the sarcolemma, which is the plasma membrane of the muscle cell. Okay? Causing the muscle fiber to contract. We'll see that coming up. Again, just to show you that we're not making this stuff up, right? Here's a scanning electron microscopic shot here of a neuromuscular junction. We've got the alpha motor neuron coming down, and here's the terminal branches coming off the motor neuron, synapsing onto the muscle cell at the neuromuscular junction. That's a beautiful shot. Here's another one that's been stained. It's not a scanning, it's just an electron microscopic shot. Again, the same thing. Alpha motor neuron up here coming down. All our terminal branches. Here are our muscle fibers, and here are our neuromuscular junctions. Another beautiful shot. Okay. Always discussed in the fractal period. Okay, in terms of neurotransmitters, in terms of the common neurotransmitters, acetylcholine. Okay, primary neurotransmitter from the motor neuron to innervate skeletal muscle, and most parasympathetic nerve. We've already heard that before. Okay. And norepinephrine is a neurotransmitter that's used obviously in the sympathetic nervous system. Okay, so parasympathetic is acetylcholine. Acetylcholine is used as the primary neurotransmitter at the neuromuscular junction, and norepinephrine for the sympathetic nervous system. Okay. Okay, that's just key points. Through those. All right, postsynaptic response. Okay, do we always, basically, if the presynaptic side, we've got neurotransmitters being released. Do we always get an action potential being fired at the postsynaptic side? No, we don't. Okay? Depends on a lot of factors. So what we got here, obviously these are our neuron, these are the dendrites, and we've got these synapses coming in, right, from other neurons. Okay? So these are what we call our presynaptic inputs. We can have positive ones, and we can have, in addition, we can have negative ones. And that depends on the neurotransmitter. So you're thinking, well, okay, what about the excitatory stuff? So it depends on the neurotransmitter. You have glutamine and acetylcholine being released from the presynaptic sides. You're going to fire an excitatory, okay, postsynaptic potential. If you've got an inhibitory postsynaptic potential, it's because the neurotransmitter being released is going to be GABA-glycine. Okay? So you've got GABA-glycine being released. In fact, you're going to take the threshold, you're going to take the action, you're going to take the membrane potential further away from the threshold. Okay? If you've got glutamine and acetylcholine being released, chances are you're going to reach threshold and find an action potential. What happens then? You've got hundreds of these coming in. Okay? And this is just showing you 1, 2, 3, 4, you've got hundreds of them coming in. Okay? All this information is, if you like, looked at by the axon killer. It's all summed at the axon killer. Because it's the axon killer that triggers the action potential. Now, neuron. So a summation of the impulses necessary to generate an action potential is monitored at the axon killer. Okay? So you've got EPSPs and IPSPs. Those are inhibitory, those are excitatory. Okay? Depending on how many are coming in, that's all summed up. And it's all summed up, then does it reach threshold? If it reaches threshold, you fire an action potential. Okay? So it's not just a case of everything, you always fire an action potential. It might be in some cases you want to inhibit the response. Okay? Okay, the book does have a fair amount on the brain. I'm not going to spend much time on the brain at all. Not in this particular lecture, okay? Just become familiar with the general regions, four major regions of the brain, okay? But beyond that, we're not going to spend much time on the brain. We just don't have time, okay? And it doesn't become that important to us. However, the peripheral nervous system becomes important to us, okay? In terms of those peripheral nerves, okay, going out, say, to skeletal muscle and receiving information, we've got a sensory division, you recall. These are sensory division, we call those our afferent nerve fibers, bringing information in from the environment. Okay? And we have our motor division, transmit information from the central nervous system by efferent, you recall, nerve fibers to target organs. As you know then, each peripheral nerve, right, carries sensory neurons and efferent neurons. Okay? In terms of the sensory division, bringing information in from the environment, right? We've got receptors we need to talk about briefly. Mechanoreceptors, thermoreceptors, nociceptors, photoreceptors, chemoreceptors. Mechanoreceptors respond to mechanical forces such as pressure, touch, and vibration. So I'm doing that. Okay, I know I did it. Okay. Thermoreceptors respond to changes in temperature. If something's hot, I know it's hot. Okay. Nociceptors respond to painful stimuli. Photoreceptors, we're not really going to worry about that in this class. It goes on to light, to our vision. Chemoreceptors, we will be talking about coming up as well. Respond to chemical stimuli from food, odor, and changes in blood concentration of gases like CO2 and O2. Monitored very, very closely in the body. Okay, so they become important to us down the road. And we also have receptors, of course, in the muscle and joint nerve endings. We've got the kinesthetic receptors in joint capsules. They sense position and movement of the joint. We've got muscle spindles. It sense how much the muscle is stretched. Hope you've seen this before. Golgi tendon orbits detect the tension of the muscle. Okay? So that provides information about the strength of that the actual muscle contraction. So you've got the kinesthetic receptors, the muscle spindles, and the Golgi tendon organs. In terms of the motor division, again, you would have seen all this before. In P215, you've got the autonomic nervous system made up of sympathetics and the parasympathetics. Again, just a reminder, what's the sympathetic nervous system? What's the main neurotransmitter? That's right. Norepinephrine, right? And for the parasympathetic, then it's serotonin. Okay? They antagonize each other. They're different, right? They obviously always function together. In terms of the sympathetic system, nervous system, you've heard the term probably the fight or flight response. Okay? Prepares the body to face crisis. So right before I came into class, my heart rate was elevated, right? Fight or flight response. Increased heart rate. You can read all this without me doing it. Okay? Vasoconstriction in active vascular beds. This is the predominant nervous system when you're exercising. Okay? Increased glucose release in the liver, blood pressure, bronchodilation. We'll talk about that coming up through gas exchange, etc., etc. And of course, the parasympathetic nervous system is going to be the complete opposite of this, right? Decreased heart rate. Now, obviously, me just standing here at rest at the moment, the parasympathetic nervous system is predominating, right? Keeping my heart rate down. So that's our housekeeping or vegetative state, if you like. Okay? Opposed actually those of the sympathetic nervous system. That's just how we look there. We've taken 3.1, the effects of those. There's the key points. Okay, without reading all that through, in terms of the sequence of events then in the sensory motor integration, okay, kind of a very simplified overview here. We're talking about stimulus to the skin. The nociceptor will pick that up, relay the information through these afferent neurons, the sensory neurons, right, through the peripheral nervous system and up, obviously, into the central nervous system. Okay. So it's going up the projection neurons into the central nervous system. We're going to evoke a response that comes back down. Descending projection neurons out through the peripheral nerve, efferent nerve fibers, and out to the muscle. Okay. In terms of those sensory receptors, this is just pretty much talking about what I just said, or showing what I just said. In terms of the different receptors, you've got your pre-nerve endings in the skin, these different receptors. You've got your muscle spindles, your Golgi tendon organs, all kinesthetic receptors for muscle, all coming in, relaying into the central nervous system, and information does go to the cerebellum, it does go into the central cortex, and then synapsing onto interneurons, goes into the motor cortex, finally the frontal lobe, then the information will come back down. Okay? Okay. You may be in the doctor's office, right? You've been hit on the leg. It's below the knee. And you get an involuntary response, boom. Leg shoots out. Does that engage the central nervous system? No, it doesn't. It's a reflex. Okay? So the doctor hits you with a hammer just below the knee. Okay? That information is picked up in the quadriceps muscle. Okay? And the information is going obviously up the sensory neuron, afferent neuron, okay, into the spinal cord. That afferent neuron, synaptopsis called an interneuron, in the grey matter of the spinal cord, puts synapses straight onto an alpha motor neuron, goes back out to the odds of the hamstring, and the leg will go out. Okay? That's right before. This is also, what it doesn't show is that obviously here, there's a synapse. In this afferent neuron, there's a synapse with a projection neuron going to the brain as well. That occurs just after, okay, this reflex. Because obviously you know you're going to be hit on the leg, right? You know, but it occurs just after this reflex. That's the first thing. Motor control, we pretty much spoken about motor control. In terms of the muscle spindles, and the Golgi tendon organs, I'm not going to go through this at length. I'm going to let you read this. So you can read through muscle spindles, okay? Golgi tendon organs. Here's a schematic in the book that talks about those, okay? I want to really get on to, again, just talk about briefly picking up in this lecture and the motor response, okay? Okay, what's important? Just a reminder. Mind you, previous classes, each muscle fiber is innervated by only one neuron. Okay? But one neuron may innervate up to several thousand muscle fibers. So what we mean by that, for instance, here's my alpha motor neuron, right? You saw before all those terminal branches coming off. Okay? They're all going to synapse onto muscle fibers. Okay? That's what that means. Each muscle fiber is innervated by only one neuron, but not one neuron may innervate up to several thousand muscle fibers. All muscle fibers are within a motor unit. That's a motor unit. Are of the same fiber type. So, this is a slow twitch neuron, which is a smaller diameter neuron. All these muscle fibers are slow twitch. Right? If that's a fast twitch muscle fiber, all these muscles, neuron, all these muscle fibers are fast twitch. What you don't see is slow twitch, fast twitch, or fast twitch A, fast twitch B, a whole mix. That wouldn't work, obviously. Okay? Motor units are recruited in an orderly manner. Slow twitch neuron, so slow twitch motor units are recruited before fast twitch motor units. Which is fairly intuitive, that should make sense. Right? Okay. Obviously, the more force that is needed, the more units are recruited. That should make sense. Okay. Like I've already said, motor units with smaller neurons, the slow twitch units, are pulled on before those with the larger ones, okay, the fast twitch. Just as an example, muscles controlling 5 movements, those controlling the eyes, a small number of muscle fibers per motor neuron, about 1 neuron for every 15 muscle fibers. Muscles with more general functions, such as those controlling the calf muscle, okay, in the leg have many fibers per motor neuron, about 1 neuron for every 2,000 muscle fibers. Okay, and that's just an example here of an alpha motor neuron synapsing onto these different muscle fibers. But remember, that's a motor unit, so all those muscle fibers, if that's a slow twitch, all these slow twitch muscle fibers. But the fast twitch motor neuron, that will be fast twitch muscle fibers. Okay? And remember the definition, motor unit is a motor neuron plus all the muscle fibers it contains. Okay? Okay, so that's the end of lecture 1. Okay, what we'll do, we'll take a break. Okay, so we'll get